This is the autonomic system physiology presented by the SHRTB with reference Color Atlas of Physiology by Agamemnon Despopoulos and Stefan Silvernac. So now here we are going to visualize a different schematic view of the autonomic nervous system. But before we start, I'm going to give a brief introduction to autonomic nervous system. So but before we go to the autonomic nervous system, so let's introduce the nervous system. The nervous system is mainly made up of two main parts. We have the voluntary nervous system and we have the involuntary or the autonomic nervous system. The voluntary nervous system is what you can control, you can feel by your will, while autonomic nervous system is done unwilling. Example, you are not capable of controlling your respiratory rate, you are not capable of modifying your heart rate, you are not capable of modifying your blood pressure. All that is being done by the autonomic nervous system. Okay, the autonomic nervous system is divided into two main parts. We have the sympathetic nervous system and we have the parasympathetic nervous system. And now, you need to point up the fact that for a voluntary nervous system, there are no need to have the ganglia. Ganglias are only formed when you have, due to the type of the neuron that you have. But for autonomic nervous system, we need to know that the neurons are never going to achieve their target, are never going to attain their target organ if they never pass through a ganglia. So as a general rule, just put in mind that autonomic nervous system are always going to have a ganglia, full stop. Now the next point is that for parasympathetic system, the ganglia for parasympathetic system are going to be closer to the target organs. Now, while the sympathetic system, the ganglia are going to be closer to the spinal cord or further away from their target organ. And now, the parasympathetic ganglia these are different points that you need to visualize. So now, the next thing that you need to visualize again on autonomic nervous system is the fact that of the origin of the autonomic nervous fibers. For parasympathetic system, the fibers all are originating from a craniosacral origin, meaning that parasympathetic fibers come from cranial nerves and from the sacral nerves, while sympathetic nerves are thoracolumbar origin. That is, all of them originate from the thoracic part of the spinal cord and then the lumbar nerves. So now all of them are going to be controlled by a subordinate center, the supraordinate center, which are going to give impulses to the craniosacral for parasympathetic and thoracolumbar for sympathetic. Now, the main facial nerves, the main cranial nerves which are involved in parasympathetic action are going to be cranial nerve 3, oculomotor nerve, cranial nerve 7, facial nerve, cranial nerve 9, glossopharyngeal nerve, and cranial nerve 10, vagus nerve. Then we have sacral nerves also involved for parasympathetic. As you can see here, cranial nerve 3 gives sympathetic to the ciliary, parasympathetic to the ciliary ganglion, which is involved in controlling the iris and the ciliary muscles, controlling the length of the tension of the lens. Then we have the facial nerve and the facial nerve are going to control glands, and the glossopharyngeal nerve control glands in the mouth. For the facial nerve is going to control the submandibular gland and is also going to control the submandibular gland and the sublingual gland. Also, the facial nerve is going to also control the lacrimal gland producing tears. We also have the glossopharyngeal nerve. Glossopharyngeal nerve controls the otic ganglion, which is going to control the parotid gland. So any secretion that comes from the parotid gland is via the glossopharyngeal nerve. Then we have the vagus nerve, which is going to control everything inside the thorax and also controlling the abdomen till the distal, till the proximal 2/3 of the transverse column. So all of them are going to be supplied: the heart, the bronchi, the gastrointestinal tract, the ureter, all of these are going to be supplied by vagus nerve. Then we have the sacral plexus that is going to supply the distal 1/3 of the transverse colon, the lower colon, the urinary bladder and the genitalia, all of them are going to be sacral, that is sacral origin. So now let's now go to the next point, which is going to be the thoracolumbar origins, that is for sympathetic nerve. So for thoracolumbar origin, everything that supplies the head is going to come from T1 to T3, from T1 and to T3 origin. So anything in the head, and all of them are going to synapse inside the cervical ganglion. You have three main cervical ganglion: the superior, the middle, and the inferior cervical ganglion at different locations inside the neck. Then inside the thorax, you have origins. You can be from T1 to T5 inside the thorax. Every sympathetic origin, every sympathetic nerve inside the thorax are from T1 to T5 origin. And then inside the abdomen, we have from T5 till T12. T5 to T12 are going to supply inside the abdomen, and lastly we have L1 and L2 are going to supply inside the pelvis for sympathetic. And now we need to know something. I initially told you that you have fibers inside the abdomen are going to come from T1, are going to come from T5 to T12. But now the question to ask ourselves is, how are those fibers going to enter the abdomen? They enter the abdomen via nerves called Splanchnic Nerve, the greater, the lesser, and the least Splanchnic Nerve, as you're going to see in the tutorial of the thorax and abdomen. So now let's visualize now this one. You have this sympathetic, you have alpha, you have beta in the eye, you have alpha in the glands, you have beta gland inside the heart, you have alpha and beta receptors inside blood vessels and all that. So all these are going to be stimulus, sympathetic stimulus all through. For the lumbar are going to be supplying mostly pelvic organs and also the adrenal medulla. So you have the bladder and you have also the genitalia supplied by the lumbar, the L fibers. So now you have cholinoceptors. The cholinoceptors, that is are going to be both nicotinic receptors and muscarinic receptors. So all post autonomic ganglia cells they are going to be nicotinic, are going to have nicotine receptors. So all post autonomic ganglia cells are nicotine receptors. Now muscarinic, all target innervated postganglionic parasympathetic nerve fiber. So all targeted innervated postganglionic parasympathetic nerve fiber. So inside the ganglia you have muscarinic receptors, while so here you need to know that what inside the ganglia you are going to have nicotinic receptors. So inside the ganglia we have nicotinic receptors. So the synaptic nerves, inside the ganglia are going to have nicotinic receptors. Now the muscarinic receptors are mostly found at the level of the target organs. So at the level of the end plates you are going to have muscarinic receptors. Now adrenal receptor alpha are mostly excitatory receptors, except at the level of gastrointestinal tract. We need to know that the parasympathetic action in the gastrointestinal tract is to increase peristalsis. So now the sympathetic action in the gastrointestinal tract is going to reduce that peristalsis. So you see that it's inhibitory gastrointestinal tract. So where you have indirect relaxant, you also have usually inhibitory. So beta are usually inhibitory, except the beta ones are usually inhibitory, except in the heart where they are excitatory. You have beta 1 mostly found in the heart, and beta 2 that are found in the bronchi, in the urinary bladder, the uterus, and gastrointestinal tract. So these are all the origins that I've initially explained that are going to come from the different nerves. So now these are acetylcholine. So now let's visualize neurotransmission in autonomic ganglia. I've initially told you that in the ganglia itself you mostly have for cholinergic receptors we mostly have nicotinic receptors at the level of the ganglia, while at the level of the motor end plate you mostly have muscarinic receptors. Now These are acetylcholine. We need to know that the presynaptic action potential are going to cause calcium ion to bind with the acetylcholine vesicles, and now acetylcholine is going to be lost. So it can bind either to the muscarinic receptor, or it can bind to nicotine receptor, irrespective if it is found to autonomic ganglia or not. So you have this. So now for nicotine receptors, you have sodium ion is going to enter, because nicotine receptor are going to be ionotropic receptors. So sodium ion enters directly and going to produce an action potential, which is early excitatory postsynaptic potential. And now for cholinergic receptor, it binds to the muscarinic receptor at this time, and now a secondary messenger made up of IP3, inositol 145 triphosphate, and diacylglycerol are going to increase the calcium ion level, and that increase in calcium ion levels are going to cause a late excitatory postsynaptic potential. And lastly, we also have the peptide, which is going to cause again another one. So this one is not mainly involved in this tutorial. So now we have cholinergic transmission in the heart. So this is how the cholinergic transmission from the parasympathetic system is going to cause inhibition or reduction in the heart rate. So what happens is that the cholinergic system are mostly inhibitory at the level of the heart. So here we have the cholinergic system. This is choline is going to enter here, and when choline enter here, we need to know that acetylcholine esterase is involved in breaking down of acetate and is going to be involved in breaking down acetylcholine to acetate and choline. So now we have now the this choline, the acetylcholine, which is going to bind to the metabotrophic receptors. Now when it binds to the metabotrophic receptor, we have both the GI protein, which is going to inhibit the adenylyl cyclase, and the GI protein is going to cause now potassium ion to move out of the cell. Since potassium ion move out of the cell, we are now going to have hyperpolarization, and now that hyperpolarization is going to cause now the reduction in the slope of this cell. So as you can see, it's going to reduce as such, and then you have negative chronotropism and a negative dromotropism. So it's going to reduce the rate of the action the rate of action potential in the sinoatrial node, and also going to reduce the rate in the atrioventricular node. So these are the different things to visualize. Now we have adrenergic transmission. So this is calcium ion, this is calcium ion, this is these are the different adrenergic, the fate of norepinephrine inside the cell. So we are not going to explain that one. So these are all also how the norepinephrine are going to act. We need to know that norepinephrine is going to act on all these alpha cells. So now let's visualize the action of alpha 1, alpha 2, beta 1 and beta 2 receptor. Alpha 1 is going to act via GQ, and then see potassium is going to move out, and also is going to act via the activation of the phospholipase C. Activation of phospholipase C is going to cause the breakdown of phosphoinositol 1,4,5-diphosphate to produce diacylglycerol that is going to activate phosphokinase C. And also you have IP3, so which is inositol 1,4,5-triphosphate, which is going to cause calcium ion to be released. So this is going to cause hyperpolarization. So the alpha 1 in certain cells is going to cause hyperpolarization, in others is going to cause calcium ion increase. Alpha 2, you have inhibitory G and you have O. So this inhibitory cause calcium ion to increase. So you have the calcium ion is going to reduce. So you also have this hyperpolarization. So alpha 2 are mostly inhibitory. Then we have beta 1. Beta 1 activates CMP, makes more CMP to be produced, and CMP is going to cause activation of the phosphokinase A. Activation of phosphokinase A is now causes calcium ion to increase, and that calcium ion increase is going to cause activation of the cell. And beta 2 instead it also causes CMP to increase, phosphokinase A to be activated, and calcium ion to increase to reduce instead inside the cell. So now this hyperpolarization, it causes the alpha 1 causes inhibition in gastrointestinal motility. So you need to know that alpha 1 is involved and can be causing hyperpolarization, which causes inhibition of gastrointestinal tract motion. Then this alpha 1 also can also cause contraction in blood vessel, contraction in bronchial, contraction in sphincters, contraction in uterus. Now the alpha 2 receptor, is going to cause it has both two fates. You have one where you have calcium ion reduction, another one where you have hyperpolarization. So all of them are going to be inhibitory to the salivary gland. So it inhibit the salivary gland, inhibit insulin release, inhibit norepinephrine release, and inhibit acetylcholine release. Then we have beta 1. Beta 1 is going to drive the heart, causes more heart to contract, the heart rate to contract, and also cause renin release. And we have beta 2. Beta 2 now causes instead the dilation of the vessel, dilation of the bronchioles. So you need to know that alpha 1 causes contraction of the bronchioles, also with the parasympathetic, but beta 2 instead causes dilation of the bronchiole, dilation of the uterus, dilation of the vessels or blood vessels, while alpha 1 instead causes contraction of blood vessel, contraction of bronchioles, contraction of the uterus, but beta instead causes dilation. So the alpha are contraction and beta 2 dilation. So from here, say thanks for your kind attention. This is the end of Autonomic Nervous System tutorial. So say thank you, and please don't forget to like and subscribe for the channel. Thank you.